Here are the proteins, and these are membrane-bound proteins. Proteins are made of chains of amino acids, actually very long chains of amino acids. We can do other things with amino acids, but proteins are long chains of amino acids. Proteins are manufactured by ribosomes, and proteins are machinery of the cell. Proteins have all sorts of words. They'll end in things like globulin or globin, as in glob or globe. Like immunoglobulin is an immune globulin. It's made by the immune system. It's made by lymphocytes. And globulin means it's a protein. Hemoglobin, heme, is the iron-containing heme group in the center of it. Hemoglobin is the stuff that makes your blood red, carrying oxygen to the cells, carrying carbon dioxide away. Please don't forget that when someone else asks you that. Hemoglobin is a protein. Protease, anything that ends in ase, as in ASE, is a protein. Like a protease is something that cleaves a protein or a peptide bond. So protease is a protein that cleaves a peptide bond. Transcriptase, we're going to transcribe the plans for DNA inside of the cell using a protein called transcriptase. Kinase, I've mentioned before, is a protein that's involved in phosphate transfer from the word kinetic. So anything that ends in ACE, anything that ends in globulin, anything that ends in globin, those are all proteins. When we talk about receptors, we're talking about proteins. When we talk about enzymes, we're talking about proteins. When we talk about ribosomes, we're still talking about proteins, okay? There's going to be free proteins. Those float around either intracellularly or extracellularly. But important in here, membrane-bound proteins. Those are made by ribosomes that are attached to endoplasmic reticulum. So membrane-bound proteins are made in the rough endoplasmic reticulum. During the course, I will draw my membranes just like this. Extracellular, outside of the cell, is always on top of the membrane on the pictures, and intracellular is always on the bottom of the membrane. So there's no confusion. That is a membrane-bound protein. I think I mentioned that. And free proteins are not attached to membranes. Let's talk about some proteins that I'm sure you've heard before. The sodium-potassium pump. The sodium-potassium pump uses ATP to push sodium out of the cell and put potassium into the cell. And what this does is create a capacitance, an electrical capacitance across the membrane, essentially creating a battery across the membrane. So some things can use the energy of ATP without even metabolizing ATP. They can use sodium as a driving force. And so we're going to talk more about that. The sodium-potassium pump creates an electrical gradient across the membrane. It's okay. Sodium-potassium pump is a protein. That's what I wanted you guys to know. The reason I put these little gears here is the protein changes shape and ratchets and moves to move the little sodiums out of the cell and the potassiums into the cell. And so it's supposed to show that there is a mechanical component going on. There's a chemical signal that's going to activate a membrane-bound protein and let sodium back into the cell. And it'll have a fancy name like a ligand-gated ion channel. And we'll talk about those during neurotransmission. But when we talk about chemical signals signaling receptors to open ligand-gated ion channels, whatever that means, we're talking about proteins because that's what this portion of the lecture is about. Or a chemical signal can activate an enzyme. A chemical signal meets its receptor and activates a chemical reaction to occur inside of the cell. And so here, what I'm trying to show you is this chemical signal came from another cell of the body to tell this cell of the body to do something. We'll talk about that with endocrinology. And here, that enzyme is metabolizing a chemical reaction, and the chemical reaction here, some square thing being transformed into a triangle. It's certainly more complicated than that, but that's what I wanted to show you. So enzymes are proteins that facilitate chemical reactions by lowering the activation energy required. You can read all about that. Of course, it can be far more complicated than that. There's my chemical signal activating a receptor. You'll read about G-coupling proteins if you read about things too closely in this course. All right? And here we've activated adenylyl cyclase, and adenylyl cyclase will take ATP and convert it into a second messenger. Here we are multitasking with ATP. Right? Yeah, of course there's energy transfer involved, but the ATP is being converted into something called a second messenger, which is now a signal inside of the cell. And so we've activated the enzyme adenylyl cyclase to create a second messenger inside of the cell. We're going to transport glucose. When we talk about insulin and diabetes, we're going to need to transport glucose into the cell, and glucose is going to do that through, glucose is going to get into the cell by facilitated transport, and that transport system is what? A protein. And by the way, we pump amino acids into the cell. Because of the phospholipid bilayer, glucose can't just diffuse into the cell. Amino acids can't just diffuse into the cell. They have to be transported into the cell using proteins. So acetylcholinesterase, anything that ends in the word ase, is an enzyme, a protein. Okay? And what acetylcholinesterase does is break down acetylcholine into acetate and choline. I will talk about this more again, I promise. Okay? And it's a protein. Proteins are made of chains of amino acids, very, very, very long chains of amino acids.